Okay, does that sound okay? All right, great. So I'll be talking about multivoxel pattern analysis, which is a relatively new or newer method for fMRI data analysis that can tell us in a bit more detail about the representations that the different regions that Nancy's been talking about employ. Okay, so as Nancy sort of described extensively, we've learned a good amount about the functional organization of the human brain from fairly straightforward methods of fMRI data analysis, so just looking at which brain regions respond more to certain categories of stimuli than others. However, we might want to know a lot more than just what certain brain regions respond to, and in particular, we might want to know what dimensions of the stimuli the regions are actually representing. So for instance, I've two images of faces on the right here, and if you measure fMRI responses in a number of subjects in face regions to these stimuli, you'll probably get pretty similar responses. The face regions tend to respond quite strongly to just about any face that you present, and don't differentiate strongly between different faces. However, if we think that this region is representing certain dimensions of these faces, such as aspects of facial features or face identity, presumably there are some differences in the underlying neural responses in these regions to these two face images. And the thought behind MVPA is that maybe if on top of that there's actually some spatial organization to those differences in underlying neural responses, we might actually be able to pick up on that difference by looking at spatial patterns of fMRI responses across the region, rather than just averaging the response across the region in a sort of standard analysis set. Okay, so just to give you a sense of the sorts of features that I'm thinking about in these types of analyses, particularly for category-specific regions like the ones that Nancy's been talking about. So for face regions, you might think that they could represent aspects of specific facial features like eyes, noses, mouths, etc., facial identity, head orientation, gaze direction, expression, and so on. For scene-selected regions, so regions that respond specifically to scenes, some hypotheses are that they might be involved in representing the identity of a specific place. They might be involved in representing scene categories, so whether it's a beach or a forest or so on. They might be involved in representing the geometric layout of the scene, your orientation within that scene, and so on. And for body regions, you might think that they're representing something like the specific body part that you're looking at, features of that body part like the shape or muscle tone, or the configuration of a full body, so the structure of a body. Okay, so I'll just dive into kind of specifically how we're going to do this. So I'll talk first about sort of the most basic type of analysis that is used in MVPA. The basic idea is to see whether there's a consistent difference in the evoked pattern of response across two conditions within a certain region. So basically, you're going to scan a number of subjects while they view a certain number of stimulus categories. You're going to split your data in half, generate activation maps for each category in a given region. So for instance, let's say we have one category that's visual images of chairs and another that's images of cars. We're going to take the first half of the data and extract evoked patterns of response to the two categories. We're going to also do this in the second half of the data, the point of this being to sort of get a sense of our noise limit and how consistent the patterns of response are just within a given category. We're then going to compute correlations between these patterns of response. So we'll compute these within category, again, to just get a sense of how reliable these patterns are and what our noise level is, and compute correlations between categories. And basically, we're going to ask whether there's a difference between the within category correlations and the between category correlations. The idea being that if there's a reliable difference, that indicates that there's some information in the pattern of response in this region that differentiates these categories. Okay, so again, we'll do some sort of statistical comparison of the within and between category correlations. And if there's a significant difference between those, we'll claim that there's information in the pattern in this region that can discriminate those categories. Okay, so I'll walk you through first just one study that uses this method that I think is a particularly nice example of a way of getting at which features a region represents about a certain type of stimulus. So this is a stimulus known as a point light display. It was introduced by psychologists named Johansson in the 70s. The kind of cool thing about this stimulus is that you can get pretty detailed information about the nature of the motion of this human agent. So you can see that he's doing a jumping jack, and also the form of his body, so you can see where his arms and legs are, in spite of the total lack of static body information. So you're just looking at dots that are placed at endpoints of the limbs of this agent. So if you want to know something about where in the brain this type of stimulus is processed, you could do a sort of standard fMRI analysis. So presents animations depicting point light displays of human motion, like the one I just showed you, and compare that to point light displays depicting object motion. So for instance, just a rigidly rotating or translating object. If you do that comparison, you get something that looks like this. This is a sort of large meta-analysis looking at 121 subjects' responses. So you get activation in a pretty broad swath of temporal cortex. And in particular, there are sort of two foci. So one, which turns out to correspond to the extrastriate body area, which, as Nancy mentioned, also responds to static images of bodies. And one that's a bit more superior. It's called the PSTS, or posterior superior temporal sulcus. So you might want to know, then are these regions doing different things in processing the stimuli? And just with this sort of standard univariate analysis, we don't really know anything about that, right? We just know that these two regions both respond to human motion over object motion. There's sort of not an obvious next step in terms of sort of standard fMRI analyses to get at what might be different about what they're doing. Okay, so Van Genoyten et al. In 2014 tried to use MVPA to get at this question. So they presented subjects with 4 different types of point light displays depicting humans walking. So the point light displays are either going to be facing toward the left or toward the right. So there's the stimuli that are in the left-hand column and the right-hand column. And they're also either going to be walking forwards or walking backwards. So the PLDs on top are walking forwards, and the ones on the bottom are walking backwards. And they're basically going to try to see whether patterns in these two regions, EBA and PSTS, can discriminate across these two dimensions. So the body form or body orientation dimension, so leftward or rightward facing, and the body motion dimension, so walking forwards versus walking backwards. Okay, and the way that this sort of split half analysis that I described before will play out in this specific case is that we're going to, again, split our data into 2 halves, so the first and second half of the data. And to look at form information, we're going to collapse across the motion dimension. So we're going to collapse across forward and backward moving point light displays, and just separate data based on whether the walker is facing to the left or facing to the right. We're then going to, as I described, compute correlations across the 2 halves of the data in patterns of response in these 2 regions, both within category and between categories, and see if there's a significant difference between those. If there is, we're going to claim that the patterns in this region have some information about body form or body orientation. Okay, and likewise for motion, we're going to collapse across the form dimensions, so collapse across leftward and rightward facing, separate the data in terms of whether the point light walker was going forward or backward, and then again do the same sort of comparison of within condition correlations to between condition correlations, and if there's a significant difference, we'll claim that there's information and patterns in this region about body motion. Okay, so I'll just show you the results. This study used sort of a separate scan to localize these 2 regions, both the EBA and PSTS, and then looked at patterns of response to these 4 conditions within those regions. This is showing results from the EBA. So what I'm plotting is for the form index, the difference in correlations within category and between category for the form separation, and the motion index is the same thing, but for the 2 sets of motion conditions. And basically what you can see is that there's significant discrimination for the form index, but not for the motion index. So it seems that EBA patterns discriminate between walkers that face to the left and face to the right, but not between walkers that are moving forwards and backwards. And in contrast, in PSTS, you get actually the opposite pattern results. So here, patterns seem to discriminate between walkers that are moving forwards and backwards, but not those that are facing left and right. So the argument here is that in the EBA, there's some representation of the orientation of a body, and in PSTS, in contrast, there's some representation of the specific kinematics of body motion that agents under. Yeah.
They look in V1 or MT?
Yeah, so they look in MT and don't find either discrimination. They don't look in V1. But yeah, one thing I didn't mention is that they do, so they're having subjects fixate gaze, and they do vary the positions of the presentations a bit, so you wouldn't expect to get it in V1. All right, so one other important thing to ask, so you can imagine, so say in this experiment you just had 2 conditions, so say just the leftward and rightward facing walker both moving forward. If you were able to decode orientation in that sort of experiment, you might want to claim that that discrimination was about the heading orientation of the actor, But in fact, it could also be about just low-level stimulus compounds. There are similarities in just the individual motion trajectories of specific dots in the animations, and that could be driving this sort of discrimination. So an important thing for MBPA studies in general is to demonstrate some sort of invariance with respect to other stimulus properties. So this study nicely has this 2x2 design, so it can directly look at this. So it can ask for the form information. Does this generalize across the two different motion conditions, and vice versa for the motion information? So in this analysis, instead of just splitting the data in half in a sort of standard way, we're going to split it in half across the orthogonal dimension. So to look at form information, we'll split it by the forward and backward moving walkers, and then again do this sort of standard comparison of within category and between category correlations. And for the motion information, now I'm going to split by form information, so split the leftward facing and rightward facing walkers, and again look at within versus between category correlations. And indeed, they find that the EBA has form information, so its patterns can discriminate the rightward and leftward facing walkers, even when you're generalizing across different motion scenarios, so generalizing from forward moving versus backward moving walkers. And likewise, you still get this effect for the motion index in the PSDS. There seems to be pattern information that can discriminate forward and backward moving walkers, even when you're generalizing across facing left and facing right. So there's a pretty strong demonstration that this effect is not sort of driven by low-level stimulus features like renotopic position or the specific motion trajectories of individual dots, but it's actually about these sort of higher-level properties like the orientation of a body or the motion trajectory of a body. Okay, so the conclusions from this study are that EBA patterns carry information about body pose, and in fact information that's invariant to body motion kinematics. And in contrast, PSDS patterns carry information about body motion kinematics, which is actually invariant to the orientation of the body. Okay, one important thing to mention about this study, and this applies basically to MBPA studies in general, is that there are some potentially interesting null results in this study, but we don't really want to strongly interpret these null results. The reason is basically that it's entirely possible that, for instance, the EBA does actually have a representation of body motion, but that there's just not sort of a clear enough spatial organization to that representation, such that it can't be read out at the level of, a relatively low resolution fMRI signal. So if you have, sort of strong positive results in MBPA studies with strong demonstrations of invariance, you can make strong claims from that, but from negative results, it's hard to interpret the lack of a representation. Okay.
Slightly object.
Okay.
I don't really object, but it would be unparsimonious to say that the body form information exists at a scale you can see with MBPA in one region and exists but at a different scale in the next region. I mean, possible, but unparsimonious.
Unparsimonious but possible, but there are certainly examples of representations that are not spatially organized, like place cells in the hippocampus. So, if you did this before, if you tried to decode spatial position at the hippocampus using MBPA, you probably wouldn't be able to, and you wouldn't want to conclude that there's not a representation of space there, right? Okay, so just a quick summary of a few other things that we've learned about high-level visual regions using MBPA. So there's been a fairly extensive set of studies on object shape representations in lateral occipital concepts, which is a region that responds to objects over other kind of non-object visual stimuli like textures. There's sort of extensive evidence that these regions do in fact represent the shape of objects. Studies of face regions have demonstrated that there are view invariant representations of face identity in a number of these regions. So you can classify identity even subject to changes in viewpoints. The author of this is Stefano Anzalotti, who will be here at some point, so you can talk to him about this if you want. In scene selective regions, there's some evidence for representations of both scene category, so whether a scene is a forest or a room or a beach or so on, and also the geometric layout of a scene. And lastly, in the RTPJ, which is a region that seems to be involved in processing others' mental states that Nancy briefly alluded to, there's some evidence for representations of specific types of mental states. And I think Rebecca will talk more about that tomorrow. Okay, just a brief bit on different types of classifiers that can be used for MVPA. So the sort of correlation analysis that I described before is sort of the most simple way to analyze data or analyze patterns of fMRI responses. You can think of the problem in MVPA more generally in a machine learning context as a classification problem. So basically, you have a number of individual data points corresponding to individual stimuli. So for instance, individual images that you present to a subject. Your data points correspond to patterns of response to each of these stimuli. So you have a space in which each dimension corresponds to the response of a given voxel within a region. So a point in that space corresponds to a pattern of response across voxels. And basically the problem of asking whether patterns in that region discriminate to conditions corresponds to a classification problem. So you're asking whether there's some separation of data points corresponding to those two categories in this space. And it turns out that the correlation method that I just described is basically a very simple one nearest neighbor classifier where you take all your data points for the two categories and just average them to get a mean response, and then just do one nearest neighbor classification based on that mean response. But as probably many of you are aware, there are often better things that you can do than that. So some of the things that have been used in the literature are Gaussian classifiers such as LDA or SVM classifiers. And in some cases, these have been shown to do somewhat better in terms of classification. So if your goal is to kind of maximize classification based on patterns in a region, it's potentially better to move to these slightly more sophisticated methods. And one thing to note is that you might have noticed that I'm sort of only mentioning linear classifiers in this discussion. There's sort of two reasons for that. So nonlinear classifiers have typically not been used in this literature, in part because just empirically they don't seem to work very well. So in cases where linear and nonlinear classifiers have been explicitly compared, linear classifiers actually seem to do better for this sort of data. The other reason is sort of more conceptual. It's the idea that if you want to know about what a region is representing, a good thing to ask is what can be linearly read out from population codes in that region, rather than nonlinearly read out. And a good way to illustrate this is in terms of individual neurons. So for instance, if you take responses of individual neurons in the retina to a range of different images, in principle, there will be some nonlinear classifier that can do all kinds of stimulus discriminations. So, things as high level as object recognition. And we know that because that's what the visual system is doing, right? It's applying some nonlinear transformation and going from, a retinal level, set of responses and getting high level things like object recognition. So, but we wouldn't want to say that the retina is involved in object recognition per se. So, it's sort of more appropriate to make conclusions about what a region represents by asking what can be linearly decoded from that region. So that's sort of a more high-level motivation to use linear classifiers for this type of research. Okay, another question of relevance to this research is what sort of patterns can we pick up on? So in particular, fMRI is sort of a low-resolution method, as Nancy mentioned. That's one of the primary limitations of fMRI. But is it possible to pick up on somewhat finer spatial scale patterns using MVPA, which sort of takes advantage of this pattern information? And this has been asked in a number of ways, but I think most compellingly with orientation selectivity. So within the large scale retinotopic organization of early visual cortex that Nancy mentioned, there's also a finer scale organization in terms of orientation preference. So there are different regions of visual cortex that have preferences for edges at certain orientations. And this organization is sort of embedded within the retinotopic organization of visual cortex, and it's at a relatively fine spatial scale. So it's at a scale of about 300 to 500 microns. So this image is showing sort of a schematic of a patch of visual cortex where different colors correspond to regions with a certain orientation preference. And the black grid, which hopefully you can see, corresponds to sort of a superimposition of a grid of fMRI voxels on this pattern. So the lines here. And as you can see, the voxels are quite a bit bigger than the orientation columns. However, you can imagine that if there's some sort of biases in basically the number of orientation columns of a certain type within a given voxel, you might still be able to pick out, so individual voxels might still have preferences, if weak preferences for certain orientations. So this is shown here. So this voxel on the upper left responds a little bit more strongly to these orientations shown in green than the one that's shown in blue. and therefore it still might in principle be possible to decode orientation from patterns of fMRI response, even though it's this relatively low resolution measure. Okay, so Kamatani and Tong in 2005 tried to do this. They measured responses in early visual cortex, so B1 and B2, when subjects watched a number of different oriented gradings, so gradings of 16 different orientations, and they tried to build an SVM classifier to predict the perceived orientation based on B1 and B2 patterns. And they were in fact able to do this quite well. So these images on the right, in the upper left is the orientation that was shown to a given subject, and the radial plots show the predicted orientation based on this SVM. And you can see that they're doing quite well. They almost always get roughly the right orientation. Okay, so they conclude from this that orientation can in fact be decoded from patterns of fMRI response in early visual cortex, and furthermore, they sort of postulate that this is based on an ability of this relatively low resolution method to pick up on this finer spatial scale information in orientation columns. Okay, so I should mention that since this paper, whether this theoretical claim about picking up on fine spatial scale information has basically been debated for the past 9 years, and it's actually still a matter of debate. Tong and other groups have been going back and forth on this for quite a while. But I'll just give you a brief account of sort of where that debate has gone and the current status of it. So the major complaints about this was that it turns out that there's actually also, in addition to the fine scale orientation column organization, a sort of weak large scale orientation preference across visual cortex. So what I'm showing here is just the standard retinotypic organization of B1 and B2. So the different colors correspond to regions that have preferences for stimuli in a certain angular position within the visual field. And it turns out that if you torture your subjects and scan them for almost 2 hours looking at oriented gradings in different orientations, you can actually find a similar map of orientation preference that's sort of overlaid on this angular position preference in early visual cortex. This was done by Jeremy Freeman and David Eager's lab. So the claim of this paper is that, in fact, orientation decoding could be just based on this large-scale orientation map, and it could not be picking up on sort of the fine spatial scale information that's known to be present in orientation columns. Okay, and the most recent stuff on this is actually so recent that it's not published, so unfortunately I don't have figures to show you on this, but the claim is basically that, in fact, you can sort of model out this large-scale orientation preference and still do above chance classification of orientations from B1 and B2 patterns. So that suggests that these large-scale patterns might be playing a role in the original paper, but you can actually get rid of them and still have some information about orientation, suggesting that you do get some of this fine fit spatial scale structure in MVPA responses. And also sort of particular interest, you can find individual voxels that have stable preferences for multiple orientations, which suggests that those preferences can't be just driven by this large-scale map. That would just predict a single preference for a given voxel. But that paper is yet to come out, so we still have to evaluate that, see if we think it's true. But there's at least some hope that some of the patterns that we're reading out in MVPA could be driven by these very fine spatial scale patterns. Okay, so I don't have too much time, but I'll talk a little bit about representational similarity analysis, which is a somewhat more recent tool that sort of provides a more full or rich description of the representational space of a region. So the idea here is to not just look at patterns across two or a small number of conditions, but to look at patterns of response in a region to sort of a large number of different categories of stimuli, or actually individual stimuli themselves, and look at the full matrix of correlations between patterns of responses to those stimuli, and to use this to sort of reconstruct a similarity space of that region, to sort of get at the features that this region might be representing. And actually the standard way to visualize this in literature is not actually a correlation matrix, but a dissimilarity matrix, which is often just computed as 1 minus the correlation between patterns in a region. So an example of that is shown here. So this is a region that responds similarly to different images of houses, and dissimilarly to different images of, dissimilarly to images of faces and houses. All right, and motivation for this sort of method is that it just provides sort of a more full description of what a region's representing. You can try to do things like figure out what features of a larger space a region cares about, or what features modulate patterns of response in that region. You can try to do things like dimensionality reduction and see if there's sort of a low-dimensional space that can account for similarity patterns in this region. You can do things like structure discovery and try to find what the structure of similarity patterns in that region reflects. So is it something like a tree structure, something like a ring, something like a grid, so on. And the really nice thing about this method is that you can also use it for cross-modal comparisons. So this representational dissimilarity matrix, or RDM, abstracts away from the patterns themselves. So you can therefore use it not just with human fMRI data, but also with other modalities. So you can do this with monkey electrophysiological data. So you can take a set of population recordings in monkeys to a large number of images, correlate responses across different neurons, and use that to build up an RDM for a region of monkey cortex. You can also get an RDM out of a computational model. So you can build a computational model that applies some process to some set of stimuli, and then extracts responses, for instance, in a neural network model, at some set of high-level nodes in the model, correlate those across stimuli and get an RDM through that. You can also do this with behavior. So you can, for instance, ask people how similar a set of stimuli are, or derive this through some other behavioral measure. So you can use this to compare similarity spaces across a range of different modalities, across species, across neural recordings, models, and behavior. So it's a potentially very powerful tool for sort of integrating these different modalities. So I'll just show, this is sort of the first example of an implementation of RSA from Nico Kriegas-Korte, who sort of came up with this term and has been popularizing this method over the past few years. So in this study, he's going to compare RSAs for a large number of visual objects in both human and monkey inferotemporal cortex. So basically object processing cortex in humans and monkeys. And with humans, he's doing this with fMRI. With monkeys, he's doing this with population electrophysiological reportings in IT. And he uses a set of 100 images that are categorized along a few lines. So animate versus inanimate, human versus non-human, body versus face, and so on. And this is the RDM for human IT based on fMRI responses. You can see there's a very strong organizing feature of this response, which is the animate-inanimate distinction. So generally, if you take two images of animate things, they have a fairly similar pattern of response across IT. If you take an animate thing and an inanimate thing, they have a fairly different pattern of response. You can also see some sort of meaningful variation within the animate square. So faces and bodies have somewhat different patterns. Right, if we compare this to the RDM acquired in monkey IT using electrophysiology, you see a surprisingly similar, or not surprisingly, but strikingly similar pattern of results. So again, you see this very strong manipulation by animacy. And in fact, if you correlate these, you get a correlation of about 0.5, which is highly significant in this data. So I don't know, I find it quite striking that you can use these totally different methods in different species and end up with a similarity space that seems quite similar. And one thing to mention is that the correlation is not just driven by the animate-inanimate divide. So you can look, for instance, just within the inanimate stimuli, and there's still a significant correlation. Likewise within the animate stimuli or in the off-diagonal block of animate to inanimate. Okay. I think I will actually have time for this. So I was just going to give one last example of sort of a cool use of RSA for a neuroscientific question. So the question here is, where can we find in the brain representations of gaze direction in perceived faces that are invariant to low-level image properties like head orientation? So humans are pretty good at discriminating the gaze of others. I think you'll probably hear from Danny Hariri at some point, and Tao Gao, and they'll tell you more precisely how good we are. But for instance, if you look at these middle images, you can sort of roughly tell that all of these are looking directly at you, despite quite a substantial variation in the specific image you're receiving based on variations in head orientation. So this study is going to present subjects with all of these different images of faces with different gaze orientations and head orientations, and ask if there are regions whose RDMs reflect basically differences in gaze direction that are invariant to differences in head orientation. So we can build an RDM for gaze direction similarity. As this block diagonal structure just based on the way that these stimuli are arranged from left to right. We can also build RDMs for low-level image properties that we want to sort of abstract out. So we can build an RDM on the top here just based on pixel-wise image similarity. We can also build one based on head pose. These RDMs are going to be correlated with the gaze direction RDM, but not perfectly correlated. So they're picking up on slightly different information. All right, so this example is going to use something called the searchlight method. So basically, instead of looking at a predefined ROI at what patterns can discriminate, we're sort of going to look across the brain for where local patterns have RDMs that fit these theoretical RDMs corresponding to gaze direction differences. So basically, at each voxel of the brain, we're going to put a little sphere around that voxel, consider the patterns of response to all of the images within that sphere, build up an RDM for each of those spheres, and then compare those RDMs to these theoretical RDMs. Okay, so this is the result of that analysis. So here, we're basically, for each voxel, computing a local RDM and correlating that with the theoretical gaze direction RDM. So voxels that show up in yellow here basically have significant correlations with the gaze direction RDM. You can see two regions of the superior temporal sulcus, both posterior and anterior, where the RDM appears to reflect differences in gaze direction. Furthermore, we can look for regions that in which the RDM relates to the gaze direction RDM, even after we account for effects of low-level image features and head orientation. So here we're going to be looking for regions where the RDM from fMRI patterns is correlated with the gaze direction RDM, even after partialing out these low-level RDMs. And basically, it appears that this anterior STS region still correlates with the gaze direction RDM, even after we partial out these low-level features, whereas the posterior STS RDM essentially does not. The conclusion here is that the anterior STS actually has a representation of gaze direction that's invariant to low-level image features as well as head orientation, and that this representation can be actually read out with patterns of fMRI response. Okay, that is pretty much all I had to say during time, hopefully. Just to summarize what I've been going through, so MVPA can potentially go beyond what we can conclude from sort of standard fMRI analyses and tell us about what a region is actually representing. This region's, these patterns may reflect sub-fMRI voxel resolution patterns, and it's really exciting that we can sort of potentially pick up on those, but in some cases it also may not reflect that, so you have to be sort of careful about making conclusions about this. Lastly, representational similarity analysis can provide sort of a more rich picture of the overall similarity space of the region, and can be used sort of very powerfully to combine data across different species, different modalities, and across neural data as well as computational models and data here. Okay, so that's all I have. Okay, any questions?
Okay, I'll ask one. So basically, I think it's really nice demo of the method. I have no question on that.
Mm-hmm.
And you see the STS pretty much shows up in both biological motion and social attention, like the gaze direction.
Mm-hmm.
We also know that it's pretty much involved in language processing. So it seems like this region is like really a common, and that's probably not just this region. It's probably the whole brain is more like a distributed network. So I don't know how informative is it, like the selective approach is to understand the brain. It must be really hard.
That's a very good question. So actually, we've asked this question specifically about the STS. So we looked at responses to faces, biological motion, language, mental states, and voices. And it turns out that you actually can find highly selective regions within the STS for those things. So you can find a region that responds to biological motion over object motion, but doesn't care at all about language, doesn't care about faces versus objects, doesn't care about voices, and doesn't care about mental states. You can also find one for mental states. You can find one for voices. You can sort of find one for language, although there's some modulation by mental state content. And interestingly, at least in our data, you can't find one for faces, so you get an overlapping response to faces and voices. But there does seem to be substantial selectivity even within the SGS.
And actually, so Deej, on your particular example of overlap between language and some social stuff, Ben's data actually does show some overlap in there between those two, which is something that's a matter of ongoing investigation. So I think the picture is that there really is quite a bit of segregation, but there's also some patterns of overlap, and both could be important.
Yep. Anything else? Yep.
Do people ever look at structured models to look at representations in the brain?
Yeah. Yeah, that's a great question. Yeah, so if you look at some of Kriegeskort's papers, he does some explicit comparison with neural RDMs to sort of a wide variety of models. So what exactly do you mean by structured models? Like,
Well, I don't know exactly, but it seems like you're talking about different representations in the brain, but
right.
I think you're looking at dimensions or maybe like variables.
Alex will talk a bunch about this kind of thing.
Okay.
But something that you definitely can do is you can generate RDMs from any kind of really richly structured model. Only predictions that have similar different objects will be. And then you can compare those to the neural data. You won't be actually like directly using the model on the neural data, but you can make predictions about what.
Don't you think? I mean, I thought Ed Boole had did a bit of sort of with like using like fancy non-parametric Bayesian models to analyze neural data. Or is that? I think that was one of your papers.
Well, we did this clustering thing, which is much simpler, right?
It's basically a GMM. It's basically a Gaussian function model.
Yeah, I mean, that's cool, but it's different. I mean, you're asking if you have a particular computational model, can you use that structure to query where in the brain you're going to find this thing? And Alex will talk about some methods to do that, but also, as you just said, this RSA method is another way to do that. You could get a representational similarity space out of your whatever model it is, and then look for that representational similarity structure over the brain.
Right. Yeah. I don't think they're great examples of actually explicitly doing sort of a search for structural.
You need like a good model, right?
Sure, but things like applying the Kemp-Tenenbaum model for discovering structural form to RDMs, right? That could easily be done. I think, like things that people have done are just apply something like NDS to an RDM, and just sort of look at the structure that you get in sort of a high variance subspace. And I don't know, I guess you're not talking about here. If you look at regions that respond to color, in some of those regions you'll see a structure that looks like a ring, suggesting that they're sort of representing color space. But this hasn't methods for sort of searching for the structural form that underlie RDMs haven't been applied too extensively. That's definitely an issue.
One is discovering the structure in the RDMs, in the neural RDMs. The other one is having more structured models and analyzing it.
Yeah.
You can do both.
Yeah, and that I guess we'll talk about.
This is important uninformed and maybe answer a question. So Nancy in the beginning said very nicely what you believe could or could not be done with such methods, and you built on that. So my question is, if you're the luckiest researcher alive in 10 years from now, how far do you think you could push this method?
That's a great question.
How far do I think I can push this method? So I feel like that's really an empirical question. It depends on to what extent these representations are spatially structured. Depends on how far we can push these methods in, say, 10 years. Ideal situation.
Or I guess what you're saying is how much do the limits of the basic signal limit how far we will ever go? It's a very important question.
Yeah, it's a very good question. So, yeah, I mean, there are fundamental limits on the resolution that you can obtain in fMRI data. Basically, your signal just decreases as you decrease voxel size. And we can get around that to some extent with things like higher field strength, but only to some extent that sort of introduces other problems. So I'd say, something like 1 millimeter or a bit below 1 millimeter is probably as far as we're realistically going to be able to get with fMRI. So in that sense, that is going to be a fundamental limitation of fMRI, right? If there are representations that are not spatially organized at that sort of scale, at least with MVPA, we probably won't be able to pick up those. There are potentially other methods like adaptation that can.
Translating it back to what you said, what kind of questions are hope to ever answer?
Yeah, can I just add to that, which is,
Yeah.
it's a very important question. I tear my hair out about this all the time. I actually am personally arriving at the conclusion that if you're seriously interested in high-level vision, it's the study of humans, right? Like the last 15 years has been a fun lark. We discovered some stuff, and I think we're kind of hitting a wall. Not that there's nothing left to be done, but it's very much diminishing returns. And further, one of the things we have learned from functional MRI in humans is that the ventral visual pathway in humans and monkeys is damn similar. And that means, why not use real neural recordings where you can in the ventral visual pathway, where I think the answers are going to be really similar in monkeys. For other things like studying language and theory of mind, monkeys can't do it. There is no alternative, and so we just push this as far as we can take it. But where monkeys are similar to humans, we can exploit the parallel stuff that we've seen with functional MRI in order to license importing the physiology data from monkeys and making stronger arguments that they probably apply in humans. So it's not time wasted. It's just you have to be clever about the way you bring in other methods to complement the limits of functional MRI.
Do you have a? Yeah.
Slightly more positive.
Okay. Say that more loudly.
For example, one of the sort of very obvious open questions that sort of doesn't depend on the spatial resolution is, say, what's the right way of predicting the F-phase response, right? There's no good computational model that can predict the F-phase response to a bunch of different stimuli, right? And those types of questions are just, taking the overall response magnitude, even though it's pooled across many different neurons, and trying to come up with some computational model that can predict the response of an entire brain seems like an important question that people can make progress on. And it's really limited not by the spatial resolution of fMRI, but by the sort of sophistication of the actual models that you're fitting.
But then how often do you think it's going to be the case that the models are going to, that we're going to be able to adjudicate between models with a big, crude, summed mean response value of the whole damn thing?
I think it's quite plausible. So, for example, I don't think a V1 model, for example, will be able to predict the F-phase response to a bunch of different stimuli. Right?
Yeah, but if you have a bunch of
linearly, I think that the ability to detect faces is something that is probably not linearly decodable from many different types of models. But there are certain models where they will be linearly decodable. So that's a type of question you could ask that seems highly non-trivial, and is sort of an open question that really depends on the sophistication of the computational models and not on the sort of underlying limitation of the fMRI data.
So just to sort of second that, I think Jim DeCarlo spoke yesterday, and he was saying that their models match very well with IT data, but one thing he didn't talk about was they also match very well with human fMRI data. So that's not specifically face recognition, but sort of more general.
And all of the qualitative patterns that he's found, or at least supposedly, many of the qualitative patterns that he's found by measuring single units also replicate qualitatively in the fMRI data. So that the types of units in this neural network that can predict the response of single units in different parts of the brain are also the types of units that can predict the response of fMRI blocks in different parts of the brain.
That's nice. I mean, I guess the cases where I've gotten more depressive is like, you see, I sort of skipped over all this, but there's a bunch of different states responsive regions. And 8-10 years ago, I'm like, great, here's an opportunity to map out a series of computations, a whole hierarchy, let's characterize the representations in each one. And so we used the method that Ben just described, MVPA, 7 or 8 years ago, with maximal resolution, hours of data in each subject, and we could not decode, 2 different faces that were wildly different in 2 hours of data. And at that point, I just got really depressed, and I thought, okay, we just can't see the representations that we want to see. Now, since then, there are other labs where people have been able to decode some aspects of face information from some face patches. And so it's not quite zero, it's just being difficult.
That's just because it happens to be, so if you measure the responses to facial identity in a macaque, you can see that they're just not spatially clustered. So being
a response, if a unit responds to a particular face, and a monkey that doesn't predict whether a nearby unit will also respond similarly to that face. But there are things that are spatially clustered. So, for example, there's spatial clustering for head orientation. So if a neuron exhibits tuning for a particular, left-facing head orientation, it's very likely that a nearby unit of millimeter wave will also exhibit tuning for that same head orientation. So there's some things that will be spatially clustered where you can potentially measure the response to fMRI, and other things where that won't.
Yeah, and actually, in the scene selected cortex, the picture is much better. You can actually use MBPA to see all kinds of interesting stuff in those scene selected regions, much better than the face region.
But those seems like the things you can also decode out of V1.
Out of V1, that's true. That's really good.
And how much can you push with these adaptation methods?
Yeah, that sort of got cut because we ran out of time. Adaptation, just briefly, for those of you who haven't heard about this, is that, like many different measures, from looking time in infants to, single unit responses that adapt to psychophysical measures. If you use functional MRI and you present, for example, two identical faces back to back compared to two different faces, the response in the FFA is higher to two different faces than to identical faces. And that's very powerful because whenever you have any kind of measure that's sensitive to the sameness of two stimuli, you can use it to ask what that region thinks is the same. And that's the essence of characterizing representation. So it's a very powerful lever, much like the MBPA method is. But crucially, unlike MBPA, it doesn't require spatial segregation of the relevant neural populations. So it's complementary to MBPA in the sense that it doesn't require that. And in fact, we have been able to see, for example, the thing I just described, we and most other people have seen that. So you can show identity-specific adaptation for faces in the FFA, and it has nice properties like you can get it when the faces are upright, not when they're inverted, matching behavioral data showing that humans are very good at face discrimination when the faces are upright, not when they're inverted. And so functional MRI adaptation is quite, law-like and behaves itself and delivers sensible answers in the way you hope. In many cases, even when MBPA is kind of interesting. But, every method has issues, and there are issues behind MBPA as well. So, the best you can do is triangulate between all of these and not move too hard. Yeah.
I wonder if the whole enterprise is perhaps handicapping itself unnecessarily in that it seems like the problem is like trying to decode face identity, say the problem is trying to decode face identity from FFA. We know that FFA is necessary for face processing, but there's no human walking around whose brain is just FFA, who decodes faces with just using FFA. We all have fully functioning brains, which the activity is happening all the time, everywhere, and also faces are always existing in contexts, except in laboratory experiments. So I wonder if by restricting ourselves to just these spatially localized regions, are we handicapping ourselves unnecessarily?
It's a good question, and it's a really important empirical study. Video that I think everybody already seen the amazing RBC video of simulation of the FFA. Okay. Anyway, I think it speaks to this question. The important question behind it is not just the methodological one, but the necessity and sufficiency of those codes for answering the question. So let me just back up for a second, because I sort of ran out of time and I left out an important caveat. So I gave this kind of intentionally simple-minded picture of all these specific bits of brain and all these, general bits of brain. The method that Ben talked about adds an important caveat to all of that in two different senses. One is people who have used MBPA to look in each of those specialized bits. You can find information about other stuff beyond the supposed specialization. So you use MBPA to tell, for example, whether a person was looking at chairs versus bottles, just looking at the FFA. So that's an important challenge because there is information in the FFA about other stuff beyond face identity. If you take the most extreme, simple-minded view that I sort of depicted here about extreme specificity and sensible responses, well, wait a minute, then why is there information about other stuff in there? I think that's actually a very important challenge. I think we're not really through with a clear answer to that. The possible answers, like it can be epiphenomenal, right? Really what we want to know is the causal role of that region in behavior. And I wanted to mention that just to say that MBPA is not just a cool method, it's already a method that challenged the view that I put forth so far. It also challenges that view in the broader sense of suggesting that that patterns are what for the brain, and focusing on regions might be just the wrong units of analysis. So when Jim Haxby first put forth that method over 10 years ago, he was very much intentionally saying, well, so the FFA is this piece of phase selectivity in the vesicle pathway, but who cares about that piece? It's part of a broader landscape of activation, and maybe that whole landscape is the representation. Why should we focus on the peak, where there's information in the top as well? And I think that's actually a hugely important question. Like, what are the relevant units in the brain? I try to argue that the units are these selective bits, and maybe so. I think there's some arguments for that, but it's important to keep in mind that that might not be right. Maybe the relevant representations span much bigger pieces of the brain, as you're suggesting. But this is where I think the real test will be not NDPA, not even neurophysiology. It will be causal tests where you can go in and locally disrupt. And this is just beginning. It's hard and rare, and there's not very much of this kind of thing. But there's a cool recent demo. I was at the same time, probably not. Okay, so this is a patient study by Joseph Parvizi and Colleen Gross-Spector. This guy here is shown in his hospital bed where he's just had electrodes placed on the surface of his brain, and by chance, 2 of those electrodes landed right on top of his FFA. They know this because they scanned him with functional MRI in advance. They know exactly where his FFA is. The reason he's got those electrodes in there is he has intractable epilepsy that's not treatable with drugs, and so surgeons are considering taking bits out, and they're first in there with electrodes mapping out the source of that seizure. While he's in the hospital with these electrodes against his brain being mapped for the source of the seizure, they can go in and do various things like measure responses from those electrodes while he looks at different kinds of stimuli. I guess Gabrielle probably talked about that briefly. Did Gabrielle talk yesterday? No, maybe that's tomorrow. I forget. Anyway, Gabrielle Kreiman is also doing similar stuff. But these guys further have the amazing opportunity to microstimulate, put in a current right in that region, and ask what happens. This is important because it's a direct causal manipulation, not just recording, and that's what you need to test sufficiency and necessity of that region. So let's just watch the videotape.
Just look at my face.
This is the neurologist talking.
When I do this, all right? 1, 2.
This is sham stimulation.
Nothing. Okay. I'm going to do it one more time. Look at my face. 1, 2.
Okay, real stimulation.
3. You just turn into somebody else. Don't get face and then look this. My nose got saggy and to the left. I almost look like somebody I've seen before. Somebody different. That was a trick.
Okay, so that's just one little data point there. But that's the kind of data you need to test this question of the direct causal role of different patches of the brain. And we'll get specks of data over the next decade from precious little examples like this in human patients. But this is the kind of thing that particularly with optogenetics and Musmol combined with functional MRI that can be done on a broader scale of monkeys. So I think we'll really learn a lot in the next 5 years even about the causal role of different patches of brain, which will really get at this question of whether it's right to think about representations inhabiting small patches. I mean, there's a tiny little bit of cortex that was stimulated here, and you can see the effect on paper.
And there are methods to take the whole brain and throw it. You can try to do a classification and throw out boxes that aren't informative. And you can look at which boxes in the whole brain are informative. You don't need to get bigger.
I'm worried about this marathon here. Maybe we should just let people go and just, anybody who has questions should come up here. Because you guys are starting again at 4.
We can start a little later. I don't know. I don't have any.
Anybody who needs to leave, go right ahead. But, okay, meanwhile, what's your question?
Sorry, just a quick question. Is this response purely visual, or is it something that depends on what they're doing with the visual information, like if they're...
Which response? The functional MRI stuff I talked about? Or
yeah, in FFA specifically. Like, does it matter how they're using it?
Yeah, no, it's a great question. Does it matter what you're doing with the face? Yeah, so we did all kinds of experiments, identity, discrimination, this task, that task. To a first approximation, it doesn't matter. If there's a face landing on your retina and you're not, dead, your FFA is turning on. That's not quite true. You can modulate it by attention, as I mentioned. If there's a face in the same place and you're attending like crazy to another part of space, you will damp down that response, but not shut it out. But it doesn't depend on visual representation, visual presentation strictly. You can close your eyes and imagine a face and selectively turn on your visual face area. Or you can imagine a scene and selectively turn on your significant place area. But I would say in that case, you are top-down constructing a visual representation, and it lives, it knows where it should live in the brain.
I saw a paper about using transcranial magnetic stimulation to perhaps activate part of the brain. Will you be going into that as well? Will it help establish causation?
Absolutely. Yeah, I had that in my talk, and I took it out because there wasn't time. So yeah, I take your, the question is whether transcranial magnetic stimulation is helpful as a causal test of the role of different regions of the brain. The answer, absolutely. It has, does everybody know what TMS is? Anybody want me to do 30 seconds on TMS? Right, you stick in a current in a coil like this next to your head, it makes a brief magnetic transient, it disrupts neural activity underneath the coil. You can now position that coil with respect to functional MRI data in the same individual. So unfortunately, the fusiform face area is too medial. The signal doesn't reach in there. I know, first thing I did when I found the TMS device 10 years ago, and more than that, was stick it right there, crank it to the max, try to make myself diagnose it. No, that didn't work. FFA is too medial. Many people have tried that, very disappointing. But David Pitcher did his PhD in England a bunch of years ago, and had a smart idea that in addition to the FFA, there's another face region that's out on the lateral surface, just asking for it. So you can position the coil right over that region and zap it. And Pitcher showed this astonishing result where there are three regions out there. There's the occipital face area, nearby is the exercised body area that I talked about, near that is another region that hasn't come up yet much called the lateral occipital complex that seems to be involved in processing object shape. David showed a triple dissociation with the causal test of TMS, zapping each of these regions one at a time, showing that when you zap the occipital face area, you disrupt face discrimination, having no effect on body or object discrimination. You zap the body area, you mess up body discrimination, no effect on objects or faces. Zap the object region, you mess up objects, you have no effect on faces and bodies. So those are the kind of data that, to me, suggest that even though there's NMDA pattern information, there are other things besides the preferred stimuli in each of those selective regions. Those regions are causally, nonetheless, very selective. Now, no method is perfect, especially TMS. Effects are weak and small, and there's still room to argue that you can only pick up the big effects with TMS, and a weaker causal effect might be there, but you don't have the statistical power to pick it up. So it's consistent, it fits nicely, but one still wants more data.